Associate Directors of the Social and Behavioral Sciences Core. Also on the call today is Amy Cornelli, who is the Core Director. She is a faculty member in the Department of Population Health Sciences and Medicine, and also is a faculty at DCRI. Two of our other co-directors will be presenting today, Christina Meade, who is a faculty member in the Department of Psychiatry and Behavioral Sciences, as well as secondary appointments in global health and psychology and neuroscience. Christina's background focuses very much on HIV risk behaviors in adults, particularly with substance abuse and psychiatric disorders. The third associate director, in addition to Christina and I, is Sarah Wilson, another presenter this afternoon. Sarah is a faculty member also in the Department of Psychiatry and Behavioral Sciences, and is an investigator at the VA Center for Innovation to Accelerate Discovery and Practice Transformation, and is a clinical psychologist. Sarah's work focuses on testing and implementation of interventions for substance use, psychological symptoms, and health behavior in at-risk populations. And our fourth presenter today is Janet Huebner. Janet is a senior research analyst and has been the manager of the Biomarkers Core facility in the School of Medicine since its inception, working with Dr. Virginia Krause since 2010. And Janet has significant expertise in molecular markers of multiple matrices, species, and diseases, and assists with developing informative biomarker panels to phenotype complex human populations. On behalf of the Social and Behavioral Science Core, as well as the Center for AIDS Research, our charge is to help the broader Duke community increase their impact of their HIV and AIDS research. This year, the CFAR is in its 16th year, and thanks to the leadership and contributions of so many in our community, we have another 5 years of prosperous activities ahead of us, and we thank Dr. Weinhold, one of the co-directors, for joining us today. In the SBS Core, if I can, we offer a variety of activities, and we want to support all of the members of the Duke community. And we help with grant review, manuscript development, we host think tanks. We also aim to help support people engaging in the community. And so one of our activities is Rocket Talks, and they're a new format for us that we're wanting to kind of launch momentum for new activities that relate to the social and behavioral sciences. And today we're going to focus on the value of biomarkers in social and behavioral sciences. And we have four extraordinary, well-versed colleagues here at Duke University to lead that. First of all, Christina Meade is going to talk about the value of biomarkers in social and behavioral sciences, and then talk about some of her experiences with her research about specimen collection in the community. Janet will then follow and discuss biomarkers shared resources across Duke so that people have a better understanding of the resources that are available to them. Also presenting today is Dr. Rosa Gonzales-Guarda from the School of Nursing. Rosa is also the the co-director of the Community Engagement Core for the CTSI, and her program of research focuses on immigration stress and acculturation among Latinx immigrants, and she has real life experience from the field with her project, SER Hispano. And then closing out is Sarah Wilson, who will talk about her research in the community and some of the COVID adaptations that have been implemented. And so Christina, will turn it over to you, and I think you are able to drive your slides. And if people have any questions, feel free to type them in the chat. And after each speaker, we'll maybe do one or two and then end with kind of a summary for all of our speakers to respond to any questions. So Christina, thank you for being with us.
Yeah, thank you, Michael. So I want to, so our goal today is to provide kind of a high level overview of why as social behavioral scientists, you might want to consider adding biomarkers to your project. And then, so we kind of want to just get the juices flowing. And then I'm going to provide an overview of some things to consider, like the types of biomarkers that are reasonable to collect in the field. So meaning that you're in either a community site or you're working at a research site at Duke that's not directly affiliated with clinical services. So I'll kind of walk through with things that are available and give some examples from my own research, which is primarily focused on addictions, but really we just want to get the juices flowing and let you know that we are also here for you as you start to think about biomarkers to identify what works for your research. So why biomarkers? I think while basic and clinical scientists have been using biomarkers for a long time in their research, there's now increasing appreciation that they can be valuable across sort of the larger spectrum of disciplines, and that to a large extent that they can also help increase the scientific rigor of your project and the reliability. So whereas we are experts in behavioral data collection and recognize the value of self-report, sometimes biomarkers can help provide those sort of hard outcomes to support your research. And you know, to the extent that self-report might not be perfect, there are biomarkers available that might help boost your concurrent validity of self-report. And I would argue that in a lot of cases, it's really the complement of these biomarkers with self-report that provide the most robust data point for your projects. So I think there's a lot of different ways in which it can enhance our research projects. So, I mean, for one, it can help us really establish the social and behavioral factors that are underlying disease processes. So it can really bolster the main mission of what we do as social behavioral scientists. But a lot of us also do intervention work, and there I think the biomarkers can help us to establish the impact of the intervention. And to a large extent, biomarkers can help us establish what sort of the biological mechanisms underlying processes are or the intervention. But which biomarker to include, if any, is really the million-dollar question, and it kind of depends on your specific research question as well as your budget, of course. So I'm gonna I prepared a few slides to talk about both rapid point of care tests that can easily be implemented in the field, if you will. And then I'm going to talk about blood specimen collection, because there's a whole host of things that you can get from blood data. So one of these, so breathalyzers are one type of device that can help us. So there's, sorry, step back. There is a lot of information that's available in exhaled breath that we are all learning a whole lot about in the era of COVID, right? So there are like hundreds of biological molecules that sort of come out in breath. And so breathalyzers can help us capture that. I am an addiction scientist, and so we most commonly use breathalyzers to get a sense of people's blood alcohol concentration. And there are hundreds of these types of devices that are really inexpensive and easy to use in a laboratory setting and easy to transport. They cost anywhere from $100 to a couple $100, and you can get information about whether someone has has recently used alcohol. We tend to use it to ensure that people are sober over the course of the study. So wanting to make sure that they're sober at the point of consent or before doing tests like neuropsych testing. But if you are doing an alcohol intervention project, for example, you might want to know, you might utilize it as an actual data point as opposed to a screening measure. I know that there's a lot of smoking research that's being done in people with HIV as part of our core, and carbon monoxide can be measured also through very similar breathalyzer devices. And then there are fancier devices that are coming out that are allowing us to assess for recent use of other drugs of abuse like meth or cocaine in the lab. These are a little bit more complicated than a simple breathalyzer device, but provide a lot more information as well. But I think that with this in particular, breathalyzer is just one piece of the puzzle. And this is an example of where collecting different types of data really in the end is going to strengthen it. You're not going to be able to do much with a breathalyzer without also asking about someone's use of alcohol. I think the biggest challenge, so I'd always thought of breathalyzers as being super easy to implement, totally non-invasive, but now in the era of COVID, they are actually a pretty high risk intervention. And Sarah is going to talk at the end about some really innovative adaptations that she and her team have come up with to enable the use of this technique, even with the risk of exposure to respiratory infections. So I think of great relevance to those of us in the HIV field. There are lots of rapid diagnostic tests that can be implemented. So for HIV in particular, there are both oral swab as well as pinprick tests that can give information about whether someone has HIV antibodies in their body. So we have used these kinds of tests both as measures of prevalence in particular populations. So we've recently finished a respondent-driven sampling project with one of the goals to be to estimate the prevalence of HIV in people who actively use cocaine in the Durham area. And so this test was really helpful for being able to do in a non-clinical setting. But we also use it in our own research where we want to compare people with HIV and people without HIV. We tend to use it as a sort of a confirmation of HIV negative status. A lot of time people living with HIV will be aware of their infection, and we tend to not put them through another test if we already know that they're infected, but having the confirmatory negative is important if you're doing a comparison study. So again, I'll self-disclose that I've always been a little bit scared of blood. I'm one of those people that has a severe vasovagal reaction and faints at the sight of blood. So I would always try to avoid blood. And when had the choice of using an oral swab versus a pinprick test, we used the oral in our team. But then I wanted to do HCV testing for another project, and that involves a pinprick test. And once I got over my initial concerns, I very quickly realized that they are just as easy to implement as the oral swabs. And thinking about it again with the lens of COVID, this might be a good time to choose a pinprick test over one that involves unmasking. Some things to consider though, like with the HIV tests in particular, are that these diagnostics really are just preliminary screens. There is the possibility of both false positives and false negatives. I think the positives are more common and more concerning. So it's important to be able to have a second test to offer on the spot, ideally from another manufacturer, so that if it was a false positive, you can get an answer pretty quickly. But we also have had several experiences of false negatives. Actually, our very first person in one of our tests, a recent study, someone who we know has been HIV positive for decades tested negative on our screen. And it turns out that if someone has been on ARVs for a long time, it is possible to get a false negative. So again, just another example of where biomarkers can be super useful and enhance rigor, but self-report, medical record, and other measures are important. And it's important to sort of take it all, to consider it all together, triangulate. I should mention that for a lot of these, for a lot of infectious diseases like HIV, HCV, syphilis, these are all reportable conditions. So as you consider implementing these, it's important to work with your IRB to determine whether or not you're going to have to do reporting to the health department.
Sarah, we have about a couple more minutes for your.
Oh, okay. All right. Well, so then let me just go really quickly through urine drug tests, another test that's super easy to implement in the lab and can provide from addictions to other kinds of medical conditions, but you will only get information about what's happening at that moment in time. So you will need to complement that with other self-report data. Blood. So there is so much information that you can get from blood that is relevant to HIV research. So whether it be co-infections, information about disease status, disease progression, immune activation, there is so much that you can get. So the question is, how do you think about getting blood specimens if you are working in the field? So I guess I'll just sort of get to the punchline, which is to say that it is all very possible to do. We were able to convert in our space, we converted an old kitchen into a biosafety level 2 lab space where we were able to draw blood. It doesn't take very much room. You just need a phlebotomy chair. Of course, staff on site who are trained in phlebotomy, but it is all very feasible to do outside of a clinical setting. Then you'll need to think about which tests you want to, which tests you're interested, what data you're interested in getting, and figure out what the best approach is. So for us, we decided in one of our projects that we're doing pretty standard laboratory testing. And so we set up a contract with Quest to do the testing for us. So rather than, we decided that we didn't want to send it to a Duke lab, which would involve placing orders through the Duke system and uploading things into the Epic. We decided to do a service contract instead with a private company. And it's worked out very nicely because they come with a courier service and pick up the blood after we've done the initial processing, and a couple of days later, we'll get faxed our result. So if that's something that of interest, I would be happy to talk more about how we got that set up. But for other studies, of course, you might want to think about using the Duke health system. But for other less standard tests, you are going to want to think about collaborations with investigators or core services like the one that Janet is about to tell you about. So I think that is a good time to turn it over to Janet. Unless there, I don't know, Michael, if we have time for questions, or we just want to save them to the end.
I'd suggest we maybe save them to the end, because I think some questions may be answered as we go along, but they may spark other kind of corollary questions as other speakers evolve. So Janet, we'll turn it over to you.
Okay.
And again, Janet is the director of the core biomarkers facility at Duke in the School of Medicine. And she's going to share with us, as Christina alluded to, the great resources available within the Duke University system.
Thank you so much. And thanks for inviting me to present, to talk about biomarkers. It seems like you guys are really ready to launch into biomarkers, which is really exciting. In the past, I've actually worked with Rosa before. We've analyzed some of her samples from the School of Nursing. But I think it sounds like there's a lot of great opportunities for you guys to include biomarkers in your research projects, and we're happy to help you. As Michael referred to, we've been around since 2010, and we've worked with a lot of different divisions and departments throughout Duke and also outside investigators. So we have a wealth of knowledge and we're happy to share it. And with that, I'll just give an overview of the services that we provide. My disclaimer is that my primary interest in research is osteoarthritis. So my knowledge of the literature is centered around inflammation and osteoarthritis, and I'm not very familiar with biomarkers specific for HIV research, but I think, as you'll see as I go through the presentation, that all of this is very generalizable and can be applied to a wide range of research questions. So I hope it's informative. So I first want to just go through what we consider a biomarker and how a biomarker is defined. So in 1998, the NIH Biomarkers Definition Group defined it as a characteristic that is objectively measured and evaluated, and it indicates either normal biological processes, pathological processes, or as Christina was talking about, pharmacological responses to some kind of intervention. So those are the three categories that biomarkers are really useful for. And they can include anything from genomics, transcriptomics, proteomics, metabolomics, lipidomics, epigenetics, and imaging. And many of these services are available, luckily for us, through the core research facilities at Duke. Duke really has a large system of core facilities. So if you find yourself needing any of these things, they are likely available to you. But the biomarkers core facility that I manage, we specialize in quantifying biomarkers of the immune system. And that includes any protein or enzymatic activity that can be measured in biological fluids. So typically, biomarker analyses are most frequently performed in human serum, plasma, urine. We have a lot of experience with more complex matrices like synovial fluid and cerebrospinal fluid, tissue homogenates, saliva, which may be of useful to you in the field. I didn't realize you guys collected most of your samples in the field, but saliva would be a good option for you. And even we've done some study in tears. We also have a lot of experience with different species, which doesn't apply to this group, but it encompasses more common model systems like mice, rats, guinea pigs, dogs. And even here at Duke, we have access to more exotic populations like the lemurs. There's a bunch of non-human primate studies. And I personally have directed some research projects outside of Duke with a moose population and more recently with elephants. So biomarkers are being used all across every spectrum of disease and all kinds of studies, and they're really quite powerful. So the services that we provide at the Biomarkers Core Facility encompass really two main topics. So we are available for consultative services, and those are mostly centered around strategies for sample collection, sample processing, different labeling strategies, sample management, and biomarker selection. We work with a lot of really large cohorts that involve thousands of patient samples. So that's where a lot of that comes in, because if it's your first time running a study with thousands of patient samples, that's overwhelming. So we can help you with that. And we also run any kind of chemical assay that typically measures enzyme activity in biological fluids, or quantitative profiling of biochemical and inflammatory markers in all kinds of matrices using either traditional ELISAs, which uses colorimetric detection methods. And we primarily in our core use the mesoscale discovery platform. And that's a unique platform that uses electrochemiluminescence detection. And we use that system to perform either singleplex, multiplex, or can design custom assays. So for those of you who are not familiar with the mesoscale platform, or what that can provide, I didn't know how much background to give, so I'm just going to blow through this. If anyone has questions, I'm happy to answer them. But the mesoscale discovery platform is unique in that it uses electrochemiluminescent detection and pattern arrays. And what this does is it really provides a very high sensitivity assay. So it's available in a multitude of formats. They have 96 well formats that are either single spots for a singleplex ELISA, or 4 spot, 7 spot, 10 spot. And they even have 384 well plate options, which I have trouble pipetting now because I'm getting older and that's hard, but they are available. So that allows you the flexibility to look at a single biomarker or use your very limited volume to look at multiple markers from a very small sample. So the system enables you to take measurements across a wide dynamic range of concentrations in the same sample without having to dilute them, which is very useful. And currently, MSD offers assays for 390 individual biomarkers, as well as a number of multiplex panels that include either stock cytokines, chemokines, or biomarkers of angiogenesis, vascular injury, viral situations. There's all kinds of panels available. I thought I would just briefly go into what this entails, just in case anybody's not familiar with immunoassays. But this is an image of a 10-spot plate. And the way this works is that each individual spot is coded with a specific capture antibody to a specific biomarker. And then you just add your sample to the plate, your sample binds to the capture antibody, you come back with a detection antibody that has a unique sulfur tag on it. And when the plate is electrically stimulated, that sulfur tag emits a light, and then the machine can quantify the amount of biomarker in your sample. So the nicest thing about the MSD platform is that it's flexibility. I feel like I'm doing an MSD commercial, which was not my intent, but I'll just go through the options, because what's nice about this is if you are looking for a unique combination of biomarkers, the flexibility of the platform allows you to really maximize your analysis efficiency. So the VPlex platform is their standard validated kits, and those are off the shelf. They're already put together. They're based on compatible combinations, and they provide the maximum consistency and reproducibility across lots. So these are really useful for large cohort studies. Like if you have thousands and thousands of samples that are going to require, 50 different plates, you really want it to be consistent and reproducible across all the multiple plates. The UPlex platform is a platform in which you can customize the assays to include biomarkers that are compatible with one another, but that may not be available in an off-the-shelf panel. The RPlex platform is also customizable, but that uses matched antibody sets for less common biomarkers that wouldn't be available in these off-the-shelf kits. So you can build your own depending on the compatibility. And their newest platform is the SPlex platform. And that platform provides, it's a small menu. I'd say there's probably only 12 available, but they're validated kits that give femtomolar quantities of biomarker in your sample. So I just wanted to give you an idea of why I care so much about this, because in real life, it matters. So this is just an image of TNF alpha. Which is a tricky biomarker because it's very informative, but in normal humans, it's not often measurable unless someone's in a disease state. So I thought this would be just a helpful representation. So this gray bar represents the range of concentrations in a normal human, and then in a disease human, this is the range of concentrations. So it might be that a traditional ELISA using colorimetric detection is sufficient to measure your concentrations, and that's great. And there are also high sensitivity traditional ELISAs that are available. But what's unique about the MSD is because it is so sensitive and it has a wide dynamic range, you're able to measure both normal and disease concentrations from a single sample. So the less you manipulate the sample, the better. So if you were using a traditional ELISA and then you found that your concentration was out of range for your assay, now I'd have to take that sample, I'd have to dilute it again and rerun it. And that just introduces a lot of variability in your data set. And what we aim for is the least amount of variability possible, because that gives you the strongest data set for your statistical analyses. So more recently, they've developed this SPlex assay, and this picture just illustrates how the increased sensitivity that you can get from an SPlex assay. I personally have never used an SPlex assay. I have never needed to measure femtomolar quantities of anything. That being said, no one has ever asked for a biomarker that isn't readily measurable, but it is available for certain analytes if you need it. So Nicole was kind enough to share with me some biomarkers that might be interesting to this group specifically, since this is not my area of expertise. And so I just put a list of them on this slide. And on the left, these are traditional ELISA assays with colorimetric detection. And some of the ones that we have measured before or might be of interest to you are like cortisol, which is a stress marker. We've measured that in both plasma and saliva samples. Another one was these the THC metabolites. Traditionally, those have been measured mostly by gas chromatography or HPLC or even mass spec if you want to quantify them. But I did, Christina asked me about it specifically. And so last night I looked and I did find an ELISA assay that can be used in serum, plasma, or urine that can measure those metabolites. So that might be useful to you. Another one was the metabolites of nicotine. Those are readily available, can be measured from serum or urine samples. And of course, the viral infection assays, which detect the viral antibodies in a sample, are also available. Now, those viral infection ones, I'm sure you're all aware, they just give a qualitative positive or negative result. They're not measuring the degree of viral load or anything in a sample. And in the right-hand column, I just pulled from the 390 of available biomarkers currently from MSD, ones that might be of interest to this group. And they include inflammatory cytokines. There's a new combo that's called the viral combo, and that has a collection of cytokines and chemokines that are of interest. There are lots of markers of metabolism, some of which I've listed here. Obesity, which is used for metabolic syndrome and that type of stuff. And more recently, they've started to develop markers of neurodegeneration, like neurofilament light or the amyloid proteins or tau, and those are all available. So I put the website for mesoscale if you have a particular research question in mind and you want to see if that's available for mesoscale, that's fine, or you could just email me and I'll check for you. That's my go-to because it is flexible and it gives me a lot of options. But if it's not available for mesoscale, there's tons of other resources for ELISA kits of all different walks of life. So there's lots of things to consider when you choose a strategy for biomarker analysis if you're launching into this. And I thought it would be helpful to just go through them just to get you thinking about what kinds of things you need to think about as you move forward. So the first thing is the size of the cohort. You know, are you measuring 20 people? Are you measuring 1,000 people? Do they have multiple time points per subject? And I mention that because what's really important for your statistics and your downstream analysis is if you have multiple time points from a given person, all of those samples really should be measured on the same plate at the same time, because there is some variability from plate to plate, even in the best case scenario. And if you have those time points spread across multiple plates, you're really introducing a lot more variability than you'd like. And so to get the cleanest data set possible, you really want to do these kind of measurements all at once with the whole cohort all together so that you can make the best comparison possible. Another thing to consider is what type of biological fluid do you have access to? I heard a lot from Christina about field collections, and I can't imagine how that's done, but since I'm used to a more clinical environment, so your options might be limited in the field. I don't know, but we measure a lot of things in serum and plasma. Urine should be easily obtained in the field, and Salometrics makes these easy saliva kits where you just put the cotton swab in the cheek, and then you put the swab into a tube and we can, in a liquid, and we can actually do biomarker analyses in that saliva sample. So that might be a field option. The next thing is, what biomarkers are you interested in? What's going to help you answer your research question or look at the mechanisms of your research project? Single plexing or multiplexing, that really depends on how compatible your biomarkers are and what kind of sample volume you have access to. If you only collect, 100 microliters of blood in the field, you're not really going to have a large volume to play with. So multiplexing might be an option. Another consideration is what's the expected concentration in a biological fluid. I can tell you from doing this for many years, oftentimes I will get a single sample in a large cohort that has just concentrations that are way out in left field. And 9 times out of 10, that person is Hep C positive. And it just comes after the fact where you measure it and you think, wow, this person's really an outlier. And then that person's not an outlier. So under certain conditions, there are certain biomarkers that are really elevated based on infection. So it's just something to keep in mind.
And Janet, you've got about 1 more minute to go.
Okay, I'm almost done.
Okay.
And another thing is the sensitivity of the assays, which I tried to illustrate for you before. And of course, the last thing is cost. So that is a big deal in research, and somehow it has to be paid for. But there's lots of different options. So again, I mentioned where we've been around a long time. We have a lot of experience, and we love working with investigators at Duke and beyond. And I've really had a great experience interacting with people from different divisions and departments, and learned a whole lot about all different kinds of fields. So I look forward to working with you guys if you need assistance. And if you're interested in just shooting some ideas, that's fine. But if you're interested in services too, just send me an email and include like how many samples, the sample type, the biomarkers you're interested in, and I can work up an estimate. We also provide estimates for the School of Medicine core vouchers. I'm not sure if you guys utilize those, but they're really fantastic. They give up to $10,000 twice a year to investigators to use in these core facilities, mine and the other ones on campus. And they're really helpful to get pilot data for grant submissions. So if you're not using them, you should consider it. We also can provide estimates for grant budgets and such, as long as you don't call me the night before and say your grant's due at 8 a.m. So if you decide to move forward with analysis, we'll make some arrangements to get the samples here to Carmichael. I'll perform the analysis, do the QC on the data, and then I present at the end, I give you back your data and your a report that describes the experimental design, the information, measures of precision and such. And you can get your samples back. And all of that's billed through core research at Duke, so it's very easy.
Great.
So that is the end of my presentation. I hope that was useful for this particular group. It's just an overview of the services we provide, and we're happy to work with anyone and everyone. So if you need anything, just let me know.
Great. Thank you, Janet. Very informative. And now we'll turn to Rosa Gonzales-Guarda from the School of Nursing to talk about her SER Hispano study in the field and her experience with biomarkers. So thank you for being with us today.
Rosa, you're muted. Can you?
Yes. Well, thank you for the opportunity to be here with you all this afternoon and share a little bit about this project. And it's wonderful for me to see Janet, who's helped me a lot, as she mentioned, with the resources that she shared. I'm going to be sharing with you some of the reports that she referenced so that you can get a sense of what they look like. So I'm going to be talking about our SER Hispano study, which is a study looking at stress and resilience among young adult Latinx immigrants in the research triangle. And I'm going to try really hard just to focus on the biomarker components of this as much as I can to get through. I do want to, my slides are not, let's see. Oh, there we go. I do want to highlight the members of my team. We have diverse skill sets that range from people that are bench scientists to people that are community leaders, and that's been very important for us in this work. And then we have a great team of early career investigators and community engagement leaders, people in the laboratory and staff that are very connected with the community. And I share that because I think, you know, as we've alluded to, this particular study does all our data collection in people's homes or in community offices. And that engagement approach has been really important in really, you know, fostering that trust and the acceptability of even doing this in people's homes. Okay, what we're studying is the Hispanic Latino health paradox. So this idea that immigrants are healthier when they first get to this country than the general population, but this health advantage diminishes over time. And the particular piece that we're studying is really learning how the lived experience in the United States, particularly the stress around acculturation issues, how that leads to a decay in health. And previous to this study, we had been solely focused on social behavioral kinds of outcomes, looking at the substance abuse, violence, HIV, and depression syndemic. But once we learned that stress had such a strong influence on these outcomes, we started to think about how that stress how could that get under the skin and contribute to decays in health from a physiological perspective. And so it's then when we realize that we really need to think about also looking at the consequences of acculturation stress and how resilience plays a role in that from a biological stress perspective. So these are the aims. Basically, this is a longitudinal study where we're following 389 Latinx immigrants in the research triangle to really learn how chronic exposure to acculturation stress and resilience influence syndemic outcomes and also influence stress biomarkers. And we're particularly interested under aim 2 in trying to understand what specific types of acculturation stressors, like for example, is discrimination type of acculturation stressors more harmful than more stress that's at the family level? And similar from a resilience perspective, what are some of the most protective factors at the individual, family, and community level that influences our outcomes that we're looking at? And again, these are syndemic outcomes, and then we're looking at biological stress. Originally, when we wanted to do this work, we wanted to understand impact on telomere shortening, the chronic exposure of stress on telomere shortening, because at that time there was an emerging science showing that telomeres shorten at a faster rate for people that are in high stress environments. But the first time that we submitted this, we got a lot of critiques from the reviewers around our ability to be able to observe shortening in a short period of time with this cohort. And so we're following people for 2 years. And so what we did then is we revised the application to look at the mechanisms right before telomere shortening, which includes systemic inflammation and oxidative stress. So that's why we chose these particular stress biomarkers, because really we're among the first to look at stress and stress biomarkers from the perspective of acculturation stress and in this population. I alluded to our community engaged research approach, and I can go back to that if needed at some point. So this is a longitudinal cohort study of 389 Latinx immigrants, and we do all our recruitment and everything and data collection in the community. And here's our procedures. I'm gonna focus mostly on what we do to collect the biomarker sample and how they're processed and analyzed. So after we screen people for eligibility, we schedule a home visit or a visit in a community office. And that's where we do informed consent. We go through a series of self-reported measures. We ask about 270 questions that our participants very patiently answer for us. And then at the end of the visit, we collect a blood and urine sample. And then we take that blood and urine sample to Julia Walker's lab, which is part of the resources that we have available at the School of Nursing for assisting with biomarker kinds of research. And then so that the samples are stored there until we're ready to analyze that. Here's a list of all our measures. At the very bottom, you'll see the specific inflammatory cytokines that we're looking at, as well as how we're looking at oxidative stress. So we're looking at a series of pro-inflammatory cytokines and anti-inflammatory cytokines. We thought it was important, given that we're looking at resilience, the biological outcomes of resilience as well, to have some anti-inflammatory cytokines in there, as well as a C-reactive protein. And that is, the cytokines we're getting from the blood samples that we're drawing. We're looking in the urine, we're looking at oxidative stress and using F2 isoprostanes for that. So you can see a picture. This is our one of the CRCs on the study, and you can see all the equipment there they take with them to be able to do the biospecimen data collection in people's homes. Of course, now during COVID, we've stopped doing the biospecimen collection, but we have in the last couple of weeks, we did start resuming urine data collection. Because that is something that we can do with very minimal interaction with the participants. We call them, we leave the cup in front of their door, they go inside and they provide the specimen, and then we pick it up and we go take it to the lab. So this is, for the oxidative stress, we get that from samples that we take of urine, and we, it's very simple. We take it to the lab, and then we prepare aliquots of 5 mLs, and we store it there until it's analyzed. So the blood samples and the inflammatory cytokines, they were analyzed, and they will continue to be analyzed by the Molecular Physiology Institute that Janet spoke about. But this F2 isoprostanes is something that we'll be doing inside the lab in Julia Walker's lab. So here are some of the resources that I've used for this project. I alluded already to the Physiology Institute and the resources that are available at our School of Nursing. And Janet alluded to the reports that they produce. I thought they were incredibly helpful, and I just wanted to share with you all what they look like. They go through and provide an overview of what they did, and then they actually share with you some graphs of how the data looks like. And you can see that it was wonderful for for us to have processed all these biospecimens to see that there's actual data that looks kind of organized in some way, shape, or fashion. So that was beautiful when I was able to see this report. And just want to share with you some preliminary results. These are very, very preliminary. We are actually trying to resolve discrepancies in a couple of IDs that are not matching up between participants and their biospecimens. So we have about 4 of those. So these are not by any means final, but we're seeing, some sort of trend in the relationship between acculturation stress and CRP. We hadn't seen that with the other inflammatory cytokines, but we haven't added all the variables that we're looking at. So we haven't done any analysis with any of the resilience factors, and there's probably some rethinking in terms of the way we're categorizing some of these exposures to chronic conditions and also medication use that we'll need to think through more a little bit. So just some of the lessons learned. My disclaimer was that I've always felt kind of like an imposter in the biomarker space. Feel very comfortable with community engagement and social behavioral science, but the biomarker part is a new thing for us. And so one of the things that I've learned is that, I don't have to be an expert, and I wouldn't be able to acquire all the expertise needed to really be able to think through all of this. But we're lucky enough that we have all these resources available at Duke and expertise at Duke and investigators that can really definitely bring that perspective. I have found that having some additional training around biomarkers is helpful. And the way I did that was one summer I went to a genetic institute in the National Institute of Nursing Research, which was the first time I had ever even been in a lab and really got exposed to some of this. I think that the engagement approach was really critical in terms of being able to collect biospecimens and have people open to having their blood and urine collected by Duke researchers. And so I don't want to understate that. I also think it's important to, when you're doing kind of more exploratory kinds of things with biomarkers, to being very open to the possibility and the likelihood that maybe there's not one biomarker in the ones that you're collecting that have any relationships that you have hypothesized might have.
So you've got about 1 more minute.
Okay. And just to wrap up, I think thinking about when you're starting to incorporate biomarkers and you're unclear, the science doesn't support which specific biomarkers you need to include. I think having a wide variety of biomarkers is helpful in trying to think through what are the different types of mechanisms. Having different and multiple sources of data is also important. We had been collecting and freezing urine, and I think now that we've stopped with the blood collection, we are thinking of ways that we can still meet our longitudinal aims of being able to see what the influence of stress and resilience are on biomarkers, just using the urine that were collected, because that's something that we will be able to continue. So that's it. And thank you for the opportunity. I'm happy to answer questions if we have time.
Thank you so much. Very important work and very informative. And so we'll hold any questions and turn over to our last speaker, Sarah Wilson, who brings all of this to life in kind of our contemporary society of thinking about COVID and how to make adaptations in our contemporary world. So Sarah, thank you.
Yeah, thanks. So I don't have slides, which is actually great because then I can just spend about 5 minutes so that we have time for questions. So I'm really fortunate that Rosa and Christina already laid a lot of the groundwork of this. So you all know a lot about the different kinds of samples that can be collected. So I'm just going to talk about a couple of different strategies that can be used during COVID precautions. Now, of course, if you are one of the lucky ones who's marked as essential research, there is the possibility, and I'm not sure how many studies at Duke are actually doing this, none of mine are, there is the possibility of having a masked phlebotomist and a masked participant and actually doing some sample collections that way. Now, as I said, none of my studies are like that, so I'm going to talk about some different workarounds. So one of the key things that we haven't really talked about yet today is using WebEx to facilitate sample collection. So Duke has a WebEx account, and you can actually write into your IRB to use WebEx for sample collection. So the way that we tend to use WebEx is for any kind of breath sample. So if you want to get a breathalyzer for alcohol, or if you want to get expired carbon monoxide to detect levels of carbon monoxide that are associated with tobacco use, then you would have the participant do a video call with you using WebEx. Now, of course, they would need a device and an internet connection to be able to do that. So that's an important caveat, but it can be helpful to have a list of places in the community where they can get free Wi-Fi. So if they have a car and if they have a smart device, they could just drive up to that place and potentially not even get out of the car and be able to use the free Wi-Fi to connect with you. So it's important to have those, to have knowledge about how to use those kinds of workarounds. And then they would be on the screen with you, and you can help facilitate a sample of either a breath sample. You could also help them with a saliva sample using that method. And then Rosa was talking about urine samples. So that's another one that's easier to do during COVID precautions. Obviously, you probably couldn't have them do that in a public place, but you can walk them through the steps of using a urine sample. And you can either get a cup. So if you're doing, for example, urine for detecting any kind of substance use, you can actually get cups. And I believe, Janet, you mentioned Salometrics, but another company, the ones that do the Nick Alert tests, they're little test strips for cotinine. They also make a urine cup that has the tests in it already. So you can have the participant pee into the cup to detect any kind of substance that you might want to detect from their urine. And so they could actually pee into the cup off camera and then come back to the camera and just hold it up to the screen. And that's exactly what you would do for an alcohol breathalyzer or for an expired carbon monoxide. They do the test, you walk them through it, and then they could just hold it up to the screen, and you can either do a screenshot or just have your research assistant copy down the number with the date and their initials, the study coordinator's initials, whoever's collecting that sample, so that you have the date, time, the amount of, whatever exactly you're measuring, all captured there. And then one other thing that we also didn't mention today are blood spots. So blood spots can be mailed. So that's another potential way to get biomarkers. Now, of course, what Janet was talking about with the amount that you can actually do with the amount of sample that you have is very relevant for something like a blood spot. So that's sort of a 10,000-foot view of some of the adaptations that that I know of. And of course, if any of you all know of any other adaptations, we'd love to hear them. In terms of mailing specimens in particular, I actually triple checked with the regulatory coordinator for our lab. Duke hasn't required any special procedures currently for specimens that are mailed in. So even if it's a saliva specimen, if you mail the participant one of those salimetrics tubes that Janet was talking about, it's a little swab, they stick it in their mouth, chew it around, get it really saturated, put it back in the tube, and you have to double package it. But then they can actually mail it back to you. And when you get it in the lab, all of the procedures that you need, all the precautions you need to take, there's actually a city training that you can take that walks you through all of those. And then you can freeze those samples for analysis. So, you know, I was very shocked. I was thinking, like, I know we're mailing samples, but surely we're doing something extra. But in fact, we're not, and that's okay. So, so that's sort of a general overview of some of the adaptations that folks have been making for COVID. Are there any other adaptations that anyone on the call has made? Thoughts about that?
No adaptations. I just wanted to pipe in, Sarah, and say that our lab has been able to continue collecting blood despite COVID, and without actually really, the only adaptation has been wearing a mask, but given that you're already putting so many precautions in place when collecting blood, that was the only thing we had to change.
I know we only have a few minutes left, but I'd like to thank our four presenters today for extraordinarily complimentary presentations. And so we're very appreciative to each of you. I know for those that are on the line as participants, if you have questions, I know that the four speakers would be very amenable to you reaching out to get advice or to kind of talk to more about what they're doing. But if there is one or two questions, I think we have a couple of minutes to entertain a question or so.
Yeah, thanks everybody. This was really fantastic. My question is, so when you're doing research abroad, is it generally advisable to try to find capacity within that country abroad to provide biomarker research, or should we be trying to find a way to get those samples back to Duke if we can, you know, find a way to do it? Just kind of considerations, both from the NIH, which is my kind of primary funder's perspective, and from Duke's perspective. Thank you.
I think another perspective to consider, Brandon, is in-country regulations. I know some countries do not allow specimens to leave the country because they're trying to force capacity within the country. And so if you're moving into kind of international research, one of the things to consider is going to the Office of Import and Export Controls. And I can't think of that person's name right now, but is a great resource to help give guidance exactly around that and a multitude of other issues. I mean, it's a brilliant question that you asked.
Janet, has your group ever done capacity building with labs in other countries, kind of building on Brandon's question?
So from my perspective, the most important thing about collaborating with international labs is all of the biomarker analyses should be done at once at the same lab at the same time. It's really not in your best interest to measure the biomarkers, have one lab in Europe do it, one lab here do it, and then try and combine those results, because it's really muddy data. or has the potential to be muddy. There's a lot of variability between human to human, differences in pipetting, differences in lot variability. So the best course of action if you're going to be doing large analyses is to have them measured in one place. Whether that's overseas or not is up to you, but you shouldn't have different labs measuring them and then pull them all into one data set and do analyses. That's never a good idea.
Thanks.
I'd like to thank everybody again for participating today, and want to reinforce the couple of notes that have come up. This has been very helpful and very informative. And again, there are a tremendous set of resources, both within the SBS core, but across the university. And so Janet, Sarah, Rosa, and Christina, thank you for your time, your expertise, your real life, real world experiences. It helps to put it all in kind of a reality that it is doable in the social and behavioral sciences. I wanted to remind everybody that on December 10th, I think is the date, yes, December 10th, the next social and behavioral sciences rocket talk is around mHealth. And I'd also like to thank Nicole Cruz, the coordinator for the SBS core, for bringing us all together today and keeping us all organized. And she'll be sending out more information and details about our December talk. So I wish everyone a great afternoon. Thank you for your participation. And feel free to reach out to one another if you have questions. Good afternoon, everybody.
Thank you.
Thanks, everyone.
Thank you.